Hello, Afan, can you hear me? Hello. Hello, Bhaskar sir, can you hear me?
Yes, sir.
Hello, Bhaskar sir, can you hear me?
Yes, sir.
Okay, sir. Sir, still can you hear me, right? Just for checking.
Good afternoon, sir.
Yeah. Hello, Ritya. Good afternoon. Can you hear me?
Yes, I can hear you, sir.
Yeah. Let's wait for a few more minutes.
Okay, sir.
Yeah. Good afternoon, Afon.
Good afternoon, sir.
Yeah. So others are joining now, or shall we wait for a few more minutes?
Sir, I guess they will, sir.
Yeah.
I guess I'll join, sir.
Yes. Okay. So let me share my screen. Just check whether you can able to see my slides. Okay. Just a minute. So uh, my slides visible? Hello.
Yes, sir.
Okay. We can see, right? So can we refresh till the this joins? So the terms connected with genetics. Do you all remember the terms connected with genetics?
Yes, sir. Yeah, I guess you taught till back cross to us, sir.
Okay, okay, good. So list some of the terms connected with genetics. Anybody? The terms connected with genetics. Hello?
Yes, sir.
Yes, the terms, or you can type at least the terms connected with genetics. Okay, dominant traits, recessive trait. Good. Okay, mutation, allele, law of independence, or you can say independent assortment. Fine. Okay, good. Um, so just have the terms in mind to understand genetics. The terms, understanding the terms will surely help. Okay, if we cannot understand the terms connected with genetics, it is little tough to understand the concepts of, like, the principles of genetics. Okay, right, fine. Shall we start now? Can I start sharing my screen? All are ready. So let the others
Yes, sir.
can join. We just start, right? So okay. So can you see my screen?
Yes, sir.
Yes. In today's class, we will be discussing about like the Grohar Mendel's experiments on pea plants. The reason for analyzing the Mendelian experiments is the kind of hybridization experiments performed by Grohar Mendel helped us to understand the concepts of genetics, the basics of genetics. In the sense, genetics actually involves inheritance and variation, as we know. Inheritance means how characters from parents are transferred to the progeny, maybe in sexual reproduction or in asexual reproduction. How characters are transferred from parents to progeny. So that detailed study of this is actually called inheritance, heredity, whatever. And then another important thing is variation, like when the characters are transmitted from parents to progeny, the progeny shows a kind of a little variation. It may be insignificant to show any differences, or sometimes the variation is very significant. That little variation is very significant. It definitely shows some kind of changes in the progeny. Okay, let me repeat quickly. Genetics is the study of principles of inheritance and variation. Inheritance means inheritance of characters from parents to progeny. Variation means when the characters are inherited from parents to progeny, the progeny shows sometimes a very simple variation which may have some significance or which may not have any significance, but still there may be the variation is seen. Okay, fine. So how we understood, like there are in genetics three divisions. Now they say actually four divisions of genetics. One is classical genetics or transmission genetics. Okay, keep this in mind. Classical genetics or transmission genetics. In classical genetics, we will be studying, it helps us to study how the basic principles with a kind of experiments, how the basic principles were understood by the scientific community. Okay, the kind of experiments and the kind of interpretations like the scientific community gave us. So that history, based on that, the kind of experiments, the laws, the theories, okay, with that we will be understanding the concepts of genetics. So that is classical genetics or transmission genetics. It is the study of knowing the basic principles of genetics by classical experiments performed by various scientists. One. Second division is molecular genetics. In the sense, molecular genetics means what molecules are responsible for the inheritance of character. In simple, DNA, RNA, and protein molecules. So these three molecules, DNA, RNA, and protein. So they play a crucial role. Okay, so that's molecular genetics. Third one is population genetics. Population genetics in the sense, like we all are human population, we share some characters among us, and also we are different when compared to others. We are unique in certain thing. Okay, we human beings share some characters and differ in some characters. For example, I may have a disease, you may not have a disease. My blood group is A positive, your blood group is B positive. So my air lobe is free, maybe your air lobe is attached. Okay, so these are all the simple characters which we differ from each other. Okay, so like that, any character, any one gene function is compared with in a population, within a population, like a character we are comparing in human beings. So like that, all of the genetic characters, to understand that, we just do a kind of comparison. So that is called what? Population genetics. Okay, in simple, genetics is the study of inheritance and variation. Inheritance is transmission of characters from parents to progeny. Variation is the kind of, the degree in which the progeny differs from the parent. The variation is significant or insignificant, little or big, whatever. And then genetics has three divisions: classical genetics, molecular genetics, and population genetics. In classical genetics, the basics and history we will be studying. In molecular genetics, the molecular basis of inheritance, like DNA, RNA, and protein molecule, about them we will be studying. In population genetics, we have to compare the characters within a population and with other population also. Fine. So with this, now we will let into the main topic of today's class, like Mendelian experiments. As the first division of genetics is the study of history and basic principles of genetics, so first we are going to start with Mendelian experiments. Okay, fine. So can you all, are you all listening?
Yes, sir.
Yes, sir.
So shall I ask you some simple questions?
Yes, sir.
Yes,
Yeah.
sir.
What is genetics?
It's the study of
Study of
principles of
inheritance.
inheritance,
Okay, and
Variations.
very good. And then what are the three divisions of genetics?
classical genetics, molecular genetics, and population genetics.
Very good. So what we study in all of the three divisions?
In classical genetics, we study about the basic experiments done.
Yes.
And molecular genetics, we look at the molecular things happening there: DNA, RNA, and proteins.
Yes.
Um,
And in population genetics, we compare and contrast the characteristics that differentiate people and compare them within a population.
Yeah. Very good. Within also with other population. Very good. Very good. Fine. So this is what I'm explaining you for few minutes. Fine. Now. So he is Graham Mendel. So this is, see, look at a very small place. This is his garden actually. Like genetics is a very big field now. Like for every kind of aspects connected with the living organisms, we just analyze the genes. Okay, Genes only determines all the characters. So the basic principles of heredity is actually understood, experimented in this little area only. Okay, so this is the place where Graham Mendel's most of the experiments were done. Almost, you can say, all of the experiments connected with the pea plants were done with in this particular area. Okay, the Mendel's garden. Okay. Now, Graham Mendel selected, like the pea plants. See, when naming the organisms, like there are something called local names, common names, and then botanical names, okay, or scientific names. Local names, vernacular names or common names, and botanical or even scientific names. The local name in Tamil, it is Patani Chedi, okay, Patani Chedi. In English, it is pea plant. The scientific name is, can someone tell me the scientific name of the garden pea plant? It's Pisum sativum. Okay, the scientific name is what? Pisum sativum. Okay, now, like the important characteristics why Mendel selected this pea plant in the sense, actually he wanted to work in animal model only, but the monastery did not allow him to work on animal model to do breeding experiments or whatever. So he has to select some commonly available plant. So that's why he selected. And the special aspect of this pea plant helped him to work on this very easily. And note on the important aspect characteristics of the pea plant. The first thing is, it is a bisexual flower. The plant is having bisexual flowers. In the sense, monoecious. Both male and female components are present in the same flower. And then easy pollination, self-pollination, easy cross-pollination. And more than that, this is the very, very interesting thing, contrasting characters. Contrasting in the sense, like in Mendelian experiments, also we are going to see about those 7 characters, like tall, short, purple color flower or white color flower, round shape or wrinkled shape. Okay, so like that, all of the characters showed the contrasting nature. So that's very, very interesting thing. Okay, like contrasting characters. Even though the plant exhibited some of non-contrasting characters, but Graham Mendel did not select it for his studies. Okay, he just selected 7 contrasting characters. So that's the main thing which is even easy for us to understand the concept of genetics, how characters are inherited from parents to progeny. So that contrasting characters, very easy to observe, very easy to analyze. Fine. And then very short lifespan. They will grow, develop, and within like fruit, flowering, everything in 10 to 14 weeks. Means 2 to 3 months. Okay, so that really helped him, and to see the visible characters, like very few weeks. Within a week, he could able to understand the growth and development of the plant. So the shortest lifespan also very helpful for Mendel to work on this particular plant. And then, like you saw, in little space with little requirements. Okay, very basic thing. No big skill are required to cultivate these plants. Okay, just like that. With little care, it can grow. So these are, even if you find your leisure time, that's enough for them. Okay, they do not take much of your time. So all these characters. helped Mendel to discover the principles of genetics. Now, look at the stages of development from seed, germination, and then growth, then pollination, then fertilization, the flowering, and then dispersal. Okay, so these are all the basic stages of development, and all of these things, okay, like all you can observe. Everything is so obvious, we can easily observe. Now, see, Graham Mendel did pollination experiments on his own, self-pollination. What is self-pollination? So this is a bisexual flower, as we discussed earlier. Like you can see, the gynoecium and androecium, stigma, style, ovary, and then the anther and filament. So self-pollination, like transfer of pollen grains from in the same flower to the stigma, anther to the stigma. Okay, so very easy, self-pollination. And also cross-pollination is achieved by two important steps, something called emasculation and bagging. Okay, emasculation means, look at that, removal of anthers. Emasculation means removal of anthers, so which prevents self-pollination. Okay, just cutting and removal of anther. Bagging means when the anthers are removed, like to get like in the sense you can protect your flower, you can cover the flower, for example, to enhance self-pollination or to protect the stigma for some time. You can do bagging. You can cover the, after removing the anthers, you can bag it. When you get the necessary anthers of desired character, it can be transferred. Okay, so just a check, what are the 7 characters selected by what are the reasons Graham Mendel like selecting this plant? Can someone tell me? The characteristics of pea plant. Hello?
So it's bisexual,
Hmm.
easy self-pollination, and it has short lifespan,
Hmm.
easy to maintain, like
Hmm.
contrasting characters.
Wow. Very good. Very good. So these are all the 7 characters. Fine. Stages of development. Someone tell me the stages of development starting from germination.
So first it's germination, and then after a time period of growth, then pollination occurs, and then fertilization and dispersal.
Very good. Very good. Very good. Fine. So, 2 types of fertilization.
Self-fertilization and cross-fertilization.
Okay, because of self-pollination that results in self-fertilization. Very good. So, self-pollination is what?
The pollination takes place in the same flower itself.
Okay. Hmm. Fine.
And in cross-pollination, the pollen grains from one flower are going to another flower.
Okay. So, how we can support cross-pollination? What are the 2 methods?
Emasculation and bagging.
Okay. Emasculation. It is called
Emasculation.
yeah,
Okay.
removal of the means male reproductive components called anther. Bagging means
You're
covering
protecting the flower after that.
yeah, protecting. So like that, either self-pollination or cross-pollination can be done. Okay, fine. Now, what are the 7 characters selected by Mendel? So these are all the 7 contrasting character. Seed shape, seed color, pod shape, pod color, flower position, and then stem, plant height. Okay, stem height. Look at that. In seed shape, round and wrinkled, just two contrasting character. Like that in seed color, yellow, green. Pod, full, you can say inflated, or they call it as full, or constricted, you can see constrictions all over. And the pod color, green or yellow, and the flower color, purple or white. The flowering position, you can see in all the axis, along all the axis, maybe flowering can occur, or just only in the end, in the terminal end, flowering can occur. And then the stem height may be tall or dwarf. Okay?
Sir, I have a doubt.
Yes.
Sir, by terminal, do you mean that in that plant only one flower can come then?
See in terminal, see look at that. In this plant mostly in one, but in terminal in the sense there are different variations. For example, here in the axis the flowering can occur, and from here it starts and the flower is seen here. Even the floral bud starts here, the flowers are seen only here. Can you understand? Yeah,
Yeah.
even if it starts here, it is present here. It is present here. All the flowers are like this. Okay?
Okay, sir.
Yeah. Fine. So these are all the 7 characters. Look at that. All the first characters are dominant characters, like round sheets, yellow color shape, means color yellow, and then the inflated, the color of the pod green, purple flower, axial flowering, and tall stem. All the first one are dominant characters. All the bottom one are recessive character. Okay? Say, let me ask you one simple questions. If possible, you try to answer. You know variation. What is variation? What is variation?
Slight changes in DNA during inheritance.
Yeah. Differences in character. In simple is
Yes.
variation.
Yes, sir.
There are 2 types of variation in humans. What are the 2 types? I will tell you the types. You try to give me the explanation. For example, continuous variation, discontinuous variation. There are 2 types of variation. Continuous variation in character or discontinuous variation in character.
So continuous in the sense like in every generation the variation will be seen, and discontinuous means
It will stop with that one person.
no, it will still continue, but it will leave one generation gap and continue. Is it that?
Logically, okay, but scientifically, I'll tell you good understanding of the terms. But scientifically, continuous variation in the sense, for example, you take one character, height of a person. Okay, in your class, you ask the students to stand in a row from the short one to tall one. Okay, the first person may be 4.8 or 5 feet in tall. Second person 5.1. Third person 5.3. Fourth person 5.6 inch, means feet. And then 10th person maybe 6 feet. Have you seen like that? Your friends are like that only, no?
Yes, sir.
Yes, so maybe starting from 5 feet to 6 feet, you can put 50 people in order. Okay, so that is called from 5 to 7. in between there is a continuous range of differences. Okay, there is a continuous range of differences. That is called what? Continuous variation. Can you understand?
Yes, sir.
Okay, now can you give me one more example for continuous variation?
Sir, color gradient.
Very good. The race, the color basically. Okay, from dark color to fair color, you can see a variation. Yes or no? This is called what? Continuous variation, right? And then what is discontinuous variation? Can you give me some example? Look at here. Both are variations. For example, round or wrinkled. Can you see any in between range here? Is there
No,
any in
sir.
between range here in Mendelian cake here? No. Okay. Either on the one extreme end it is round or it is wrinkled. No in between characters are seen. Okay. Two opposite, two contrasting. Look at that, tall or dark, purple or white, complete green or complete yellow. No in between colors are present. So this is called what? Discontinuous variation. Can you understand this? Discontinuous variation and continuous variation. Right? Hello?
Yes, sir. Yes, sir.
Yes,
Yes,
sir.
yes.
Now. So luckily Graham Mendel got, see, in the same plant, how many discontinuous variation, how many contrasting variation. So this really helped him to, knowingly or unknowingly, he selected this plant. So this really helped him to understand, like to work on. It was so interesting for him to work on all these characters, very visible. Okay, fine. Now, like as we, like before getting into Mendelian experiment, like so the basic terms, gene, a segment of DNA which codes for a protein, and that protein controls a function. The alternate form of a gene is called allele, and then you know genotype. The genetic composition, for example, TT, the genetic composition which determines the character is called genotype. And then the visible trait. See, look at that. This plant is, the stem height is dwarf, and this is tall. Okay, very visible. Tall plant, short plant. So this is phenotype. Visible traits are called what? Phenotype. And then homozygous condition and heterozygous condition. See, look at that. Homozygous and heterozygous, in the sense two chromosomes are there. For example, this is paternal chromosome, this is maternal chromosome. Here also same type of gene we have. For example, BB. Here also B, here also B. Means same from Appa and from Amma. Paternal chromosome, maternal chromosome has the same gene, same allele. Then it is homozygous condition. Heterozygous condition, in the sense you can say different color. Okay, two contrasting genes are present, one from paternal and one from maternal origin. So this is, condition is called what? Heterozygous. Capital B, capital B. Homozygous, dominant, of course. The capital letter represents the dominant trait. And then BB, heterozygous condition, one dominant and one recessive. And then this is homozygous recessive condition. So here homozygous dominant, and here BB means it is homozygous recessive. Okay, know this. And then the kind of gem Graham Mendel did monohybrid cross, like involving a single character, flower color alone, or stem height, or seed odor shape, or seed odor color, any one character. Two plants are selected and studied. That is monohybrid cross. Dihybrid cross means two characters, okay, together. So that is called what? How two characters from parents to progeny are inherited. How one character contrast, two parents are having two different character, how it is inherited. So that is monohybrid and dihybrid process. So first, now experimentally we will get into the monohybrid cross. Okay, are you all ready? Monohybrid cross experiment. Yes? Hello, students?
Yes, sir.
Yeah.
Yes, sir.
Yeah, good. See, a cross, monohybrid cross is a cross between two pure homozygous parents having single pair of contrasting character. Okay, cross between two parents of homozygous, and then three steps are there. Okay, first, in the first step, and here it is written in sentence, and here you can have the flowchart representation also. So what are the first steps? Like he selected two plants, homozygous for tall and homozygous for dwarf. So that homozygous, to represent that, they call it as pure. Pure means homozygous variety. Okay, that's the first step. He selected homozygous dominant. Why tall? It is a dominant character. Why dwarf? It is a recessive character. One homozygous dominant character and one homozygous recessive character. So what is the main character? These are the two contrasting nature. But what is the character he selected? Among 7, what character it is? Is this
Height
about
of the plant, sir.
yeah, height of the plant. Okay, stem height. Very good. So that's the first thing. Second one, like artificial crossing. Okay, female and male parent, and of course one male parent, one female parent he selected, like male tall, female dwarf. So like that, he himself, why it is called artificial cross, in the sense he performed this experiments, he transferred the pollen grain. Okay, so that is the second step. Okay, first step, he selected homozygous tall plant, homozygous dwarf plant, and then he did the crossing. That's the second thing. Okay, second step. Now Mendel got results. He was expecting, actually, one tall plant and dwarf plant were crossed. He was expecting in the progeny 50% tall plant and 50% dwarf plant we may get. This is what he thought. But interestingly, he did not get any of, how many times he repeated the experiments, the step 1, he is getting in the first generation, F1 means filial 1 generation, always he is getting tall plant only. Okay, like he was thinking, where is that dwarfness character, whether it will come again or not. Okay, he was speculating what happened, and then he just wanted to try with cross with only the progeny. Okay, the progenies alone were crossed, parents. Okay, he did not involve the parent organisms in the second cross, he just involved only the progeny. Okay, so when he crossed the F1 progeny, he got the F2 progeny. In that, actually he saw even the dwarfness. Okay, the dwarfness. See, keep this ratio in mind. We will discuss that. I'll show you in the next slide what this ratio. In step 3, he crossed two F1 progeny, and then he got the results in this range: 3:1, 1:2:1. Genotypic ratio 1:1, 2:1. Phenotypic ratio. I'll explain you all these things. But the interesting aspect of this, he found the dwarfness again in the F2 generation. Okay, in F1 no plant were dwarf, but in the F2 generation he got dwarfness. Okay, so here is the like statistics. He collected 1064 seeds in F2 generation. See, it is not a simple experiment. We may, for looking, just for reading, it looks step 1, step 2, step 3. But how he performed, look at his dedication, the way he performed his experiments. He collected 1064 seeds from the plants. He collected all the seeds. Okay, 1064 seeds in F2 generation. And he observed, actually here, again he has to perform all these experiments. Out of these, the seed were allowed to germinate and the plant was grown, and he observed finally the characters. So not an easy thing. He has to collect the seed and follow all these things. Here we saw, no, all these things. The seed collected, and then he shown all those things, and he observed the characters after the plant grown. Okay, so very important thing. So, and then he counted the seeds, and then he was analyzing how many plants were tall and how many plants were short. See, this is the statistics. 787 plants, seeds grown to tall plant, and 277 plants dwarf plant. So this ratio only 3:1. Okay, 3:1. Okay, this is the very important significant finding of Graham Mendel, and his conclusions. The conclusions connected with this, we will come to know that. Okay, of course, all these things we will explain now. And this is the graph for the experiments, the flow chart. Like this only you have to work out for monohybrid cross. So this is the typical way to work out monohybrid cross. Look at that. The P genotypes means P represents parental generation, pure tall male plant and pure dwarf female plant. So dominant homozygous, dominant recessive tall homozygous, dwarf recessive. Like that only you have to describe the genotype. This is genotype. Genotype la eppadi represent pannirpaanga na TT. But how you have to express the TT has to be expressed, how you can say homozygous for tall. Like that only you have to describe. This is homozygous for dwarf. So the second one is gamete formation. See here, this gamete and this gamete, if it segregates like this, it forms. And then he fused F1, like the fusion, the gamete formation. See, look at that. Selfing resulted in F1 progeny, and all the progeny were heterozygous. Okay, what is heterozygous? Look at this, is homozygous for tall, homozygous for recessive, and here genotype is heterozygous, means one tall gene, one dwarf gene. What is the phenotype of all the plants? Can someone tell me? What is the phenotype?
All of them are tall.
All them are tall. So that's the phenotype. Okay, phenotype means the visible character. Fine. No, he did not say any dwarf plant here. How many times he repeated the experiments? He got the same thing only. And now these progenies were selected and progenies were crossed. So what is the possible segregation of gametes? You can see heterozygous plants only. So this is one plant and this is one plant. It is, since it is an heterozygous plant, one tall gene, one definitely one dwarf gene it has, and like that one tall gene, one dwarf gene it has. Look at that. So this, see, when Graham Mendel explained his concepts, but nobody can able to understand, like people rejected, means they cannot understand. The reason is he did not use this genotype, phenotype, all these things. Just the phenotype characters alone he shown, and he did not talk about this probability checking. Do you know the name of the square? This is the square which is called what? What is the name of the square? Punnett square. Okay, to understand the concepts of genetics, to understand the Mendelian genetics, to predict the outcome of when two parents known genotype are crossed, what will be the phenotype of the progeny. Okay, so Reginald Punnett, a scientist called Reginald Punnett, he discovered a method, a square method, to predict the phenotypic outcome of the genotype. Okay, so this square is called Punnett square. Okay, Punnett square or checkerboard. So the genotype of the plant is heterozygous when these are crossed. Like look at that. So this one and this one. Do you all know how to work out this Punnett square? Hello students?
Yes, sir.
Yes.
Yes,
Okay.
sir.
Yeah, good. So look at that. It gives the following components. Okay, when it is crossed. So this is the results he got. Like
Sir, I
if you
have
look
a
at,
doubt.
yeah.
In F1 generation, sir,
Hmm.
he took two homogynous pure tall and pure dwarf, right? How did he know it was pure tall and pure dwarf if he didn't know about the genotypes?
Here, the FP
Yeah,
generation.
how did he know it?
See, yeah,
Yeah,
see,
how did he
listen.
know it was pure?
Yeah, in your book, like in your book, in square it is mentioned. First thing, before getting into experiment, Graham Mendel selected pure breeds. Okay, like many times, for example, he selected two tall plants, crossed them, and then if many times if he selects two plants and cross it for 100 generation also only tall plant comes. Can you
Yes,
understand?
sir.
Two
Yes, sir.
plants. He first collects observation only by he observed two plants like with they were tall and he crossed. Then he got tall plants only. And then he repeated the experiment. So how many times you cross the plant? If you are getting only tall plant, it is pure homozygous for tall. Like that, he selected his plant first. Similarly, like that, he selected the dwarf varieties also. He selected tall means dominant varieties. He selected recessive varieties. Also he selected heterozygous conditions. Okay, heterozygous plant. So he categorized the plants first and then only he performed all these experiments. Okay, recorded. See, 7 years of experiments, ma, 7 years of work. Okay, okay, can you understand?
Yes, sir.
Yeah. So now look at that. So these are all the again genotype of the progeny after the Punnett square, genotypes of the progeny, and he just this is how you have to what work out. See, take this one and write it separately like TT. How many are there? Homozygous for tall, pure tall, one, and then you
Three.
can look at the box 2 and box 3. You have two heterozygous TT. This is the second genotype. And then third genotype is small t, small t. Okay, can you see genotypes? Forget about the phenotype. Just write down the genotype alone. TT. And then this one heterozygous condition, two are there. And then here you have again one homozygous condition, recessive. So this ratio is actually called what? 1 to 1. 1 is to 2 is to 1. Actually this ratio is called what? Genotypic ratio. And the phenotype. So what is the phenotype of this? Pure tall. And this is hybrid tall, heterozygous tall, and this is pure dwarf. So coming to this part, the phenotype, three portions of the plant, all three were tall. Since they have one dominant T also, the plant will be tall. So 75% were tall and 1%, 25% were dwarf. Can you understand? Let me repeat. Two parent generation plant were selected. One is pure tall, one is pure dwarf. Gametes formed and then resulted in F1 progeny. All are heterozygous, tall in nature, and then F2 progeny, like they crossed F1 progeny and got the genotypic ratio of 1:2:1, the phenotypic ratio of 3:1. Can you understand this first? Then we will go to the conclusions of this particular cross. Can you understand this?
Yes, sir.
Okay. Now, see, like this only Grahor Mendel published his results. Okay, means I'll show you Grahor Mendel's paper also, but this experiments, and of course another experiment, dihybrid cross experiment is also there. With this experiment, what can you understand? Maybe if I ask
Can
you
you
some
repeat the
simple
question,
questions,
sir?
you can answer, right? Can I ask you some simple questions?
Yeah.
Okay. What type of parents he selected?
He selected homozygous
In
tall and
F1 generation.
homozygous
Okay, fine. How? What is the phenotype of all the F1 progeny?
tall, sir.
Very good. See, what kind of organisms, what kind of parents were selected for F2 crossing? F2 generation. How?
Heterozygous.
Yeah. What kind of parent he selected?
Heterozygous.
Yeah. F1 heterozygous parents. Very good. What is the genotypic ratio?
1:2:1.
So
Pure tall
what?
1, hybrid tall 2, and pure dwarf 1.
Okay. What is the phenotypic ratio?
3:1, sir.
3:1. Okay, fine. See, all these things are clear?
Yes, sir.
Yes, sir.
Fine. Now, this experiment is to understand the principles of genetics, right? This experiment is for what? To understand the principles of genetics. What genetic principle you understood here? We can understand here.
The genes don't get mixed.
Very good. Genes do not blend. Tall is always tall. Dwarf is always dwarf. No tall, dwarf, medium height. Nothing like that. Very good. Genes do not blend. One simple genetic concept you can understand. And then, yes, anything else? Afan, what is Punnett square? Afan, are you here? What is Punnett square? What it is used for?
So,
Hello, Afan? Hello? Afan, what is Punnett square? Okay. Anybody else? What is Punnett square?
Sir, it's a
Yes,
table
sir.
drawn to
no, sir.
get
Yes, Afan. Tell me, what is Punnett square, Afan?
Sir, Punnett square is it's like graphical representation of the possible genotypes here of an offspring
Yeah.
arising from particular cross or breeding event.
Okay. So, Reginald Punnelt is a person who discovered this method, just a square, which helps us to predict the phenotypic outcome as well as genotypic outcome of any two selected parents. Parents may be homozygous, heterozygous, or whatever. So their known conditions were crossed, and we can check the predict the outcome. Okay, so that is Punnett square. Very good. So anybody else? So what else? What, see, this experiment is to understand genetic principle. What genetic principle we can understand from this apart from that?
Like even though like two pure traits are
Mm.
contrasted, only one trait is passed on.
Mm.
Yes, sir.
Okay. 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 Okay, fine. See, listen. This helped us to understand. Okay, this experiment helped us to understand two important concepts of genetics, two laws of genetics. Okay, like two laws were law of dominance. With the help of this monohybrid cross, two laws were proposed. One is law of dominance, another one is law of segregation. I will tell you what is law of dominance. See, the first wonder of Gregor Mendel was he selected homozygous tall plant and homozygous dwarf plant. Whenever he crossed these plants, always the plants were tall in F1 progeny. So he said, in a plant, okay, in an organism, sexually reproducing organism, to determine a character, there are always two genes exist. Okay, what is his first thing? Characters are determined by factors, and these factors called genes exist in pair. Can you understand this? See, listen. Let me repeat. All our characters are determined by factors. The factors are in pair, means one factor we get from Appa and one factor we get from Amma. So it exists in pair. In that, whatever the character expressed in the F1 generation, okay, whatever character, when he crossed two plants of two contrasting character, what character expressed in the F1 generation, that is the dominant trait. The character which is expressed in the F2 generation along with the F1 character, that is called recessive character. புரியுதா புரியலையா? First,
Yes,
என்னன்னா,
Savik, but
in a diploid organism, for every character there are two factors or genes exist. Adila one gene is dominant, so dominant gene expresses its character in the first generation. The characters which is not expressed in the first generation, what character is not expressed in the F1 generation? Dwarfness. So that is recessive. Okay, so like that only in 7 contrasting character, he crossed two plants and got what is dominant trait and what is recessive trait. Okay, so even in us, okay, even in, we are sexually reproducing organisms, most of our genes are of these two type only. One is dominant gene, another one is recessive gene. Okay, we also have two genes for every character. One gene we are getting from Appa called paternal gene, and one gene we are getting from Amma called maternal gene. Can you understand this first? First principle of genetics. Okay, characters are determined by genes or factors. They exist in pair. Why they exist in pair? One we get from Appa and one we get from Amma. In that, the one which expresses itself always is dominant. The one which expresses only in the absence of dominant gene is called recessive gene. Can you understand the first aspect of genetics? Yes? Yes or no?
Yes, sir.
Okay. Please read the law of dominance again, then maybe you can even more understand the second thing. Okay, second things. So when I asked the showed the chart and first asked, someone said me, genes do not blend. Correct, genes do not blend. Okay, but so in what way tall gene and dwarf gene are different from each other, and in what way both are equal to each other? Can you able to get my question? Let me repeat my question. In what way the dominant gene and recessive gene are equal to each other? In what way they are different to each other? Can you tell me? Question purida purilia? Hello students. What is the similar and dissimilar characters of dominant gene and recessive gene? In what way they are sharing equal property? In what way they are different from each other? Hello students. Nane Salidova. Can I
Yes,
tell?
sir.
Yes. Okay, listen. Maybe my answer can help you. I think you cannot able to understand the question. Maybe my answer, if I answer, you try to understand my question. Okay? See, listen. In what way it is different in the sense, in F1 generation, when dominant gene and recessive genes are crossed, parents are crossed, only dominant gene expressed in the F1 generation. It is dominant. Okay, one is dominant. It is expressed in the F1 generation. Okay? And the F2 genes, the gene actually, the characters are seen in F2 generation also. Here you can see in what way they are equal to each other in all of the generation. For example, here, what is the ratio of tall gene and dominant gene? Ratio of dominant gene and recessive gene? 1:1. Correct? One tall gene is there, one recessive gene is there. Right? Here you look into that, the genotypes. There are 8 alleles are there. 1, 2, 3, 4, 5, 6, 7, 8. 8 are there. How many tall gene are there? How many dominant means short gene are there? How many dominant and how many recessive genes are there? Tell me how many dominant and how many recessive gene? In what ratio they are present? Out of these 8 gametes, 8 alleles, 1:1. Correct? 4 plus 4. Right?
Yes,
Null
sir.
capital T, null small t. Correct? Here 1, 2, 3, 4. Small t 1, 2, 3, 4. Correct?
Yes, sir.
Yes, sir.
What it represents in the sense, see, here also it is there in 1:1 ratio. Here also in the genotype, no difference. The number of tall gene, how many times is there? That many times the dwarf gene is also there. And to make us understand this concept only, they gave us the second law of genetics called law of segregation. Law of segregation means it may be a dominant gene or it may be a recessive gene. It is differing only in the expression of the character, but not in involving in meiosis, segregated. In meiosis, what occurs? See, listen. In meiosis, what occurs? This T is separated. Okay. And this T is also separate. Okay. T is separated and this T is separated, and here also this T is separated, small t is separated, means both dominant gene and recessive gene equally segregate in meiosis. They have the equal potential to inherited to the next generation. அது டால் ஜீனா இருந்தா என்ன, டாமினன்ட் ஜீனா இருந்தா என்ன, அது மியோசிஸ்ல இன்வால்வ் ஆகுறதும் அடுத்த ஜெனரேஷனுக்கு இன்ஹெரிட் ஆகுறதுலையும் ஒரு டிஃப்ரென்ஸுமே காமிக்காது. அதோட ஆல் பேட்டர்ன்ஸ் ஆர் சேம், ஆல் ஜெனட்டிக் ப்ராப்பர்ட்டிஸ் ஆர் சேம், எக்ஸப்ட் டால் ஜீன் அவனை எக்ஸ்பிரஸ் பண்ணிப்பான் டாமினன்ட் ஜீன், அவன் இல்லாத போது மட்டுமே recessive gene will express itself. Can you understand? Genes may be tall or dominant, dominant or recessive, will equally involve in meiosis. They segregated and they can equally have the chances of inherited to the next generation. That law is called what? Law of segregation. Please read these two laws. Please read these two laws. Based on the monohybrid cross, two important principles, laws, rules of inheritance we understood. One is law of dominant, one is law of segregation. Please understand these two. Read this and I will ask you then. Hello, students. Hello.
Yes, sir.
So, can you understand now the law of dominance and law of segregation?
Yes, sir.
Okay. Fine. Let's have a quick revision. Chumma for 3 minutes. In today's class, we discussed about what is genetics, what is variation, means genetics, inheritance, variation. These are all the terms we explained. And then Grohar-Mendel's experiments, the characteristics selected by Grohar-Mendel, and then the stages of development in pea plant, and then self-pollination and cross-pollination experiments, and then the 7 characters, contrasting characters selected by Grohar-Mendel. Here we discussed about variation, 2 types of variation, continuous variation and discontinuous variation, and then some other terms like genes, alleles, genotype, phenotype, homozygous condition, heterozygous condition, monohybrid cross, dihybrid cross, the terms, and then the steps in monohybrid cross, the graphical representation of monohybrid cross, the genotypic ratio, the phenotypic ratio, the genotypes, the phenotype in F1 generation and F2 generation, and then the conclusions basically, and then two important laws helped, means two important laws understood with the help of monohybrid crosses done by Mendelian experiments. Okay, is it clear? Is it clear?
Yes, sir.
Now can I show you? See, for example, these two laws are there. Okay, what are the two laws? Monohybrid cross oda நம்ம புரிஞ்சுக்கிட்ட ரெண்டு laws என்னென்ன? What are the two laws we understood with the help of monohybrid cross?
Law of dominance and law of segregation.
Okay, actually they are called Mendelian laws. Okay, if they do, means next class we will be discussing about dihybrid cross. There we will be understanding, analyzing one law called the law of, like someone said, independence, law of independent assortment. Another third law, they are actually called Mendelian laws. Okay, they are called what? Mendelian laws. Who proposed these laws?
Rehar
Hello?
Mandal.
Hmm?
Rehar Mandal.
See, all of us will think Mendelian, Newton laws means Newton will propose. So Mendelian laws means Mendel proposed. No, Mendel did not propose any law. He just gave his results, which represented that monohybrid and dihybrid, means like most of the terms we discussed today not even mentioned in the paper of Gregor Mendel. Okay, not even they are mentioned in the paper of Gregor Mendel. But I'll show you some more persons. Actually in your book it is there in the tabular column. Mm. See, these are all the people. Okay, can you see my slide?
Yes, sir.
Hugo de Vries, Carol Correns, Carl Correns, Eric Teschmack. Okay, name of the people என்னென்ன? Hugo de Vries, Carl Correns, Eric Teschmack. See, Mendel published his results. See, Gregor Mendel, like in 1865. Okay, in 1865 Gregor Mendel published his results. When he was explaining about his experiments, I'll show you his paper also, Mendel's paper. Just a minute. You can't and I can't and nobody can easily understand his work actually. Okay, Gregor Mendel's paper. See, this is the paper published by Experiments in Plant Hybridization in 1865. Gregor Mendel, like he published this paper. He presented his research findings on Brown National History Society. In that meeting, actually, he presented. Look at that. See, in your book, I think it is there. Who coined the term genetics? Who coined the term genetics? William Batson. Genetics in the term is coined by this person. He is the one who actually translated Mendel's paper into English. This is the translated version of Gregor Mendel's paper. Okay. Now, like you just look into this paper. Introduction, like something goes on. Chumma, I just like scroll down all these things. Mendel's paper. See, look at that. It is a very long paper. Okay, very long paper. Like the experiments, 3:1 ratio, the hybrid experiments you can see. Like 7,300 something hybrids, round this many. Out of that 7,000, 5,000 round, wrinkled 1,800. Look at the ratio actually. Not we say 3:1, but look at that, 2.96:1, 2.96:1, like that, 3.01. So 3.01:1, 3:1. Okay, so why I'm showing this paper in the sense, in this paper, like all 7 characters, stem height, the pod range, in all of these things, all 7 characters, he got almost a similar ratio. Look at that, 3:1, 2.82:1, 3.14:1, position, axial or terminal, stem height or dwarf. So only these 7 experiments only he performed. And then the F2 ratio, F1 plants were crossed and got that ratio for all 7 characters he repeated. But he did not, why I am showing this paper in the sense, he did not use any of the term like genes, any of the term like genotype, any of the term like phenotype, no genetic terms used by Graham Mendel. Okay, பெருசா எந்த genetic termsமே இதுக்குள்ள இருக்கவே இருக்காது. Okay, just common plant related terms மட்டும்தான் இதுக்குள்ளார இருக்கும். Okay, but these people, after இவர் இதனால தான், like nobody could understand Graham Mendel's work, but he was so confident, so he said, my time will come. Okay, my time, today you people cannot understand what I am trying to say, but one day you all will understand. சொல்லிட்டு in 1884ல மெண்டல் செத்து போயிடுறாரு. And 1884 Mendel passed away. In 1900s only, look at that, these people, Hugo de Vries, Carl Correns, Eric Tashmak, these people called, they repeated, okay, like they just understood, they worked, and they understood, they in their similar experiment, they got that 3:1 ratio of experiments. They thought they discovered the principle. For example, Hugo de Vries, he got the same results. They thought they understood the principles, they discovered the principles, but later all together discussed lot of controversies, and finally, like these three people, like all the contemporaries, all them, like together wanted to give salutation to Graham Mendel for his work. So they only published, reworked, means rediscovered and published Mendel's work, and to honor him, they called the research findings as first law of genetics, second law of genetics, third law of genetics, like law of dominance, law of segregation, law of independent assortment. So these are all the people actually proposed the laws. Graham Mendel did his experiments on hybridization of pea plants and published only that. The ratio alone he published. But these are all the people, all these people introduced all the terms of genetics. Okay, Batson, he is Batson, okay, introduced all the terms of genetics and explained the principles of inheritance with the help of Mendelian experiments. Mendel's experiments were used as models to explain the principles of genetics, and they then only they came to know the quality of work Graham Mendel has done. So we call Graham Mendel as what? The father of genetics. Can you understand? Hello? Hello, students?
Yes,
Okay. So, like maybe in next class we can discuss about dihybrid cross, and before that So please go through your text and the terms keep in mind, and then how to work out monohybrid cross. Today I showed how to work for tall versus dwarf gene, and you take it as an assignment. Select any of the character. For example, round versus wrinkled, like RR, capital R, small r. Capital R for round, capital means small r for wrinkled. So like that, you select any of the character, you put any of the letter. Maybe flower means FF also you can put, no problem. Any letter you can take. Try to work out monohybrid cross and try to find out your genotypic and phenotypic ratio. Yes, can you do that? Chumma, it takes some 10 to 15 minutes only, not more than that. Okay? When I come to your class, you just show me whether you get this or not. Okay? Is it clear? So, any questions or anything to ask? Hello, students? Hmm? Hello? So, can I ask you some simple questions and let's finish today's class? One each. I'll ask one question each.
Yes, sir.
Okay. What is law of dominance? Anybody else can tell me what is law of dominance? Anybody else? Law of
I'm
dominance.
sorry, sir, I didn't hear your question, sir.
Law of dominance.
So, law of dominance says when parents with like two pure contrasting traits, but only one trait will get passed on to the progeny.
Oh, means one trait is expressed, but both can. Ma, listen. Both genes have the equal tendency to pass through the progeny. Okay,
Yes, but
tall
one trait will be
character.
dominant, sir.
One is expressed, that is dominant. Can you understand? Both can be transferred, inherited, have equal chances of inheritance. But one will express itself. ஒன்னோட phenotype மட்டும்தான் வெளியில தெரியும். Another one is masked. Can you understand? So that's the law of dominance. Very good, very good. So 50% correct, but little corrections. Now, so what is next person? What is law of segregation? Law of segregation. Anybody else? Hello, students.
Sir, law of segregation, like it states that every individual possesses two alleles and only one is positive to the offspring.
Uh, okay. Two alleles are there in every organism, and then it may be dominant or it may be recessive. They equally takes place in meiosis. They have the equal chances of inheritance, okay, to the progeny. Okay, so that is law of segregation. They segregate in meiosis and they can have equal chances for inheritance. So that is in simple law of segregation. Okay, fine. So see, really like little tricky. So please go through the book, and if you cannot understand anything, like you can note down your question and ask me. Okay, so Mendelian genetics, to understand that, little patience you should have. Okay, like when we discuss about the dihybrid cross also, okay, the two laws alone we discussed, we will come to the third law of genetics also. Like if we work out the 50% monohybrid cross alone we worked out, if we work out the dihybrid cross, then we can connect both of the laws, okay, first second law and third law independent assortment. So you can have some connections between the concepts which we have to understand from Mendelian genetics, okay. So please go through your book, and I will also next time, maybe in person, maybe in the class, if we have some discussion, like it will be helpful for you to understand the concepts better, okay. So very important thing to understand the genetic concepts, okay, fine. So anyway, um, thank you so much for your patience, and without working, just with the flow chart we managed. But anyway, and dihybrid cross and all, I will help you. I'll work out in the board and show you how to do that, and you also will be allowed to do that, okay. Thank you. Thanks for your patience. You can post me the questions later also whenever you're free, okay, in person also. Please note down your doubts, okay. Thank you. Thank you so much. Thank you for your cooperation. Thank you, students.
Thank you, sir.
Thank you, sir.
Thank you. Thank you.